Okay, in this video we're going to go through 17.6, which is about sex-linked conditions, just for anyone who's been absent or who wants to re-listen for revision. Okay, so these things are all in your notes. So I'm sure we remember from last year that most human females have got two X chromosomes, and most human males have got an X and a Y chromosome. X chromosomes are just longer. I'll just make a little scribble up here for you. So X chromosomes are quite long, and a Y chromosome is a little bit smaller. Oops. And it's just got it is less lengthy and it's got smaller mass as well, slightly different shape. So two X chromosomes in a woman and an X and a Y chromosome in a man. Because when this woman here comes to produce, this man here comes to produce his sperm cells, half of the sperm cells ends up with an X in and half ends up with a Y in. So we refer to the man as heterogametic in a human. Hetero means different. So his gametes are all different. Half have got an X in, half have got a Y in. Human females we refer to as homogametic, because homo means the same. Every single one of her gametes, every one of her egg cells has got an X chromosome. They're all the same in terms of the sex chromosomes, not in terms of all these others. These other chromosomes in their homologous pairs are called autosomes. This is just in humans. Well, not just in humans, it's in a lot of species. We just need to be careful because there are some questions where birds are mentioned or moths in another question I've seen, which the males have got XX and the females have got XY, but it always tells you in the question. You just need to look out for it and highlight it and make sure you don't make a silly mistake. So in terms of how human sex is determined, we're talking about sex, not gender here. If I use a little bit of space here. So our males are XY, our females are XX. So when he comes to make sperm, like I've just said, half have got an X in, half have got a Y in. I've made this far too small. Human eggs all have an X in, so effectively you can draw out a Punnett square. If I move the sperm down here, so if an X sperm and an X egg join together, you make females, and if a Y sperm and an X egg join together, you get a male baby. Okay, so that's just GCSE revision. So really, it's the sperm cell that determines the sex of the baby. Women can only ever give those X's. We did talk about last year when we did meiosis some interesting case studies where you get this process called non-disjunction, where instead of in meiosis you should have one X and one Y go to the sperm cells, and in females when she makes eggs during meiosis she should have X's going to each of their egg cells. I'm just looking over here, I've got a rabbit trying to invade me. Easy. Occasionally, accidentally, both the X and the Y go to one side of the cell, so you can get some sperm, very, very rare, with X and a Y in them. And if they combine with an egg with an X in, you'll get Klinefelter syndrome. So this process, you'll have to remember back to last year when we did meiosis, but it's called non-disjunction when things go a little bit wrong like that. So I'm not going to harp on about these now, we've looked at these already. So what is a sex-linked condition? So sex-linked conditions are any genes that are found on the X or the Y chromosome. So in the X chromosome here, you've got some regions of these two are homologous. So if you've got a gene here, sometimes it will also be found here. Not always, but sometimes. If you've got a gene found up here on the X chromosome in a woman, you would have another homologous gene on this side, another X, but in a man you don't. So you can get some conditions where if a woman has a mutation, so I drew it out in class like, just move this camera down a bit. So say for instance you've got a real gene, so you've got a dominant gene leads to colour vision, and a recessive allele, leads to colour blindness. So let's imagine you've got a woman and she's got 2 X chromosomes. If she has a mutation in one of those genes and she ends up with a colour blindness allele by accident, then it's fine usually, because she will always have the colour vision one on the other side. So the woman will end up having colour vision. But in a man, so remember this is a woman, in a man, if he gets that mutation here, he doesn't have a backup copy here. There is literally nothing. So he will end up with colour blindness. So often these genes that are found on the X chromosome, if there is a mutation to them, that effect will be seen far more frequently in men than it would in women. So how we write these out, so we're not just going to write big B, little b, nothing like a normal monohybrid cross. We would write them attached to the sex chromosome. So for this woman here, her genotype, I would write X, big B, X, little b. That's showing me she's got 2 X chromosomes. One's got a big B on, one's got a little b. I know it's backwards to that, it doesn't matter, it's just standard convention, you put the capital first, but I could have written X little b, X big B, that's fine. This man here, I would do X little b, Y. And I've got 3 options. I can either leave the Y blank, or I can say he's X little b, Y, and put a dash to show there's nothing on the Y. Or AQA let you do a 3rd option, which is to put a 0 up there to show he's got nothing on there. I prefer these 2 personally. I usually leave them just blank. Okay, so we talked about the fact that there are a couple of common sex-linked conditions. Move that back up again. None of these you need to know about at all. It's just sometimes they come up more in exam questions, particularly colour blindness and haemophilia. Duchenne muscular dystrophy, you can read about it if you want, but it's a very life-limiting condition. So people with this condition don't normally get to the age where they start to think about reproducing. So in terms of being passed on, it's very unlikely if you are affected that you'll pass it on. If you're a carrier, you might pass it on because you would never know. Colour blindness is not life-limiting, so that often will get passed on. Haemophilia in the olden days was life-limiting, so people would die very early on, or maybe in their 20s or something, so it's unusual for it to be passed on all the time. And we did some quick colour blindness tests in class. We talked about this example. Both of them have given permission for me to talk about them, so I'm not just talking about people behind the back. But a few years ago at the Grange we had the Tom and Meg Butler. I'll just use, I can probably squeeze it up here. I'll show you what we know about their family tree because they made for a really useful little puzzle. So Tom has a sister called Meg, and if I draw their family tree out, you've got a mum and dad, and I know, so this line means they married and reproduced, and then they had 2 children. Dad was colourblind, mum had normal vision. I'm just going to write normal just to save space, but I mean normal vision. Tom was colourblind, and Meg had normal vision, but again I'm just writing normal just to make life easier at the moment. So let's think about what genotypes we know about these people. So you might get questions like this sometimes, and you're asked to deduce, to figure out what genotype someone might have. So the first thing I always do is put my X's and Y's in here. So Tom is an XY human, Meg's an XX human, mum's XX, and dad's XY. Now I know Tom and his dad are both colourblind, so can you think about what alleles they must have? We're going to use the same code as we did down here. So Tom's got to have a little b, and dad's got to have a little b on his X chromosome, and nothing on the Y. They don't have a backup. Now we could figure out what genotype mum and Meg must have had. So if I just take a different colour and I'll show you how to do this. So to produce Tom, where did Tom get that Y chromosome from? He had to get it from his dad, didn't he? That's how dads produce baby boys. They give a Y chromosome. So where did Tom get this X with a little b from? Well, it must have come from his mum. So his mum must have, by deduction, she must have had an X little b. Can we figure out what was on this other one? Well, she had normal vision, normal colour vision. So she must have had an X little b and an X big B. So she passed on that to Tom. But she has this, so you would never know she was a carrier. I would still refer to this person's phenotype as normal vision. I wouldn't refer to them as a carrier. Let's think about Meg then. What do we know about her? Can you spot what she must be? How did Meg get produced? Meg got produced because she got an X from her mum in her egg and an X from her dad in her sperm. So dad had to pass on this X with a little b to Meg. That's the only possibility. That's the only thing he could give her. Mum could have given her either of those in an egg cell. But which one must it have been? It must have been big B because Meg is not colourblind. So you can deduce people's genotype by doing a bit of detective work. I show people this case study of haemophilia. So this comes up quite a lot in exam questions. Haemophilia is a condition where people are missing a protein that they need to clot their blood. So particularly, well, now they wouldn't be so bad, they can have a replacement of that protein. But when you start to study family trees in the past, you start to see people who have died very early on from bleeds, and maybe if, you know, when little kids jump off walls and play fight and things like that, and they have a bit of internal bleeding, they would die from it often. So it's caused by a recessive allele on the X chromosome, just the same as colour vision was. So describe the following people. So for each of these in the booklet, I want to know what you can tell me about them. There are 2 facts you can tell me about them. You can tell me their sex. Remember, it's always sex, not gender in biology. Are they male or female? And also, I want you to be able to tell me, are they healthy or have they got haemophilia? So pause the video and get those done for me. And then unpause when you're ready, because I'm about to tell you the answers. So this first person here is a female and she's healthy. You could also refer to her as a carrier because she's got one of each, but she's healthy. This person is a female who is healthy. This person is a female with haemophilia. This is a very unusual person. They had to get the rare faulty allele from their dad and the rare faulty allele from their mum. So these people don't exist very often. This person here is a male and he's healthy. And this person is a male and he's haemophiliac or has haemophilia. From which parent must a boy inherit his recessive gene from? Well, here's a boy with haemophilia. An affected boy, I think we're talking about here, sorry. So he's got that Y from his dad. So all of these dodgy X's have come from mum. Dads can only pass on Y's to boys. Right, let's look at this cross here. I'll show you how to draw them out. It's a nice simple Punnett chart, just like doing a monohybrid cross. Right, I've got an affected male from haemophilia, so he's going to be X, Y, and he's going to have the little h for haemophilia and nothing on that one. And he's getting crossed with a carrier female, so she's XX, and she's got capital H and little h. And then I'm going to cross them together, so they're gametes. Look like this. Pause the video if I'm going too quick, give yourself time to catch up. And then I'm going to draw a regular old 2x2 Punnett square. And I'm going to transfer the gametes down, so here's my female ones. Here's my male ones, just check we're happy with this symbol or you can write male and female. And then we're going to fill in the Punnett square just like we would with a normal monohybrid cross. So if this egg and this sperm joined together, we would get X big H, X little h, and we would have a healthy girl. If this egg and this sperm join together, we would get this really unusual situation where we've got an affected girl. This would rarely happen because the probability of these two people meeting each other in the first place is quite slim, but haemophiliac men know about it and they would probably not get to the point where they can reproduce, or they couldn't in the past, I guess. This sperm and this egg, if they join together, we've got X big H Y, so that's a healthy boy. And this egg and this sperm, that's an affected male. Okay, it might ask you a question in the exam, like what is the probability of having an affected girl? So 25%. Okay, next thing we're going to do is look at what we call pedigree charts. So a pedigree chart is a bit like a family tree. They tend to be called pedigree charts if they're for particularly rich or important people. They used to be kept so that we make sure that people from the royal families were all intermarrying with each other to keep wealth and power within families. So pedigree charts will always give you a key. Usually you get a square for males and a circle for females, and it will often tell you if they are carriers or affected or unaffected by colouring in or shading. So here we've got unaffected males, so they've got to be X big H Y, haven't they? Haemophiliac males, so they've got to be X little h Y. Unaffected females, so given that we've got carrier as a separate box, I'm going to assume that's that. And then carrier females. And this is just showing our British royal family. So Queen Victoria was a carrier. She married Prince Albert, who was unaffected, but because she carried that recessive allele, it could be passed on to some of her children. So we did get a few of these children dying at early ages. There's another pedigree chart here. Oh, just check you actually know. So these lines, that shows two siblings. So Alfonso and Victoria married and had two children. When they married, you've got this horizontal line going across here. If you want to prove a condition is recessive, then you would look for two unaffected parents who can produce an affected child, because that shows that they both must have passed on a recessive allele. So you're going to just have a little bit of practice for the rest of this sesh. So you've got this pedigree chart set of questions in your booklet on page 34. So this looks at colour vision. And then the next thing you've got to do is this thing from the textbook that I've just popped in here, a right royal disease. This is about haemophilia in the British royal family or European royal family. So that's on page 35. And then here you've got some sex-linked problems. The quality images is pretty nasty, but the questions are pretty good. They're hard, so you'll find those two a lot easier. I need you to cross two of them out for me because they're too hard. I need you to cross out number 47 and number 50, but they do get progressively harder the further you go. Okay, so that's a video explaining how the theory works. I'll go through and answer one to the questions in a bit.